We're still in chapter 22, and this is on 22.8, control of respiration. Respiratory rhythms are going to be regulated by higher brain centers, chemoreceptors, and other reflexes. Excuse me. So, neural controls involve neurons in the reticular formation of the medulla and the pons. And then we talk about medullary respiratory centers. There are clusters in neurons in two areas of the medulla that are the most important. And this is going to be the ventral respiratory group and the dorsal respiratory group. And these are going to be shortened to VRG and DRG. So first we talk about the ventral respiratory group, or the VRG. This is a rhythm generating and integrative center, and it sets eupnea. And eupnea is normal respiratory rate and rhythm. And this is about 12 to 15 breaths per minute. The dorsal respiratory group, or DRG, this group is going to integrate input from peripheral stretch and chemoreceptors. Then it's going to send information to the VRG neurons. Not much is known about this group of neurons. Then we have the Pontine respiratory centers located in the pons. Neurons in this center are going to influence and modify the activity of the VRG. What they're going to do is they're going to smooth out that transition between breathing in and breathing out, and vice versa. It's also going to transmit impulses to the VRG that's going to modify and fine-tune breathing rhythms that occur during things like speaking, sleeping, exercising. Now, I didn't ask any questions on this, okay, because it's not yet fully understood. So don't worry about it if you don't understand it, because there are no questions on the exam about it, but I did want to at least mention it. So this is the generation of respiratory rhythm. Again, it's not fully understood. One hypothesis is that the pacemaker neurons in the VRG control intrinsic rhythmicity. The most widely accepted hypothesis is reciprocal inhibition of two sets of interconnected pacemaker neurons in the medulla generates the rhythm, and each neuron set controls the other to ensure the rhythm. Again, I'm not going to ask any questions about this on the exam. Excuse me. Now we're going to talk about factors influencing breathing rate and depth. So depth is determined by how much the respiratory center stimulates the respiratory muscles. The greater the stimulation, the greater the number of motor units excited, and this is going to increase the depth of inspiration. The rate is determined by how long the center is active, and both are modified by changing body demands. Respiratory centers are affected by chemical factors, influence of higher brain centers, pulmonary irritant reflexes, and the inflation reflex. Excuse me. Now, the most important factor of all are the chemical factors. And these are going to be changing levels of PCO2, PO2, and pH. Those are going to be the most important. The most potent one, though, is going to be PCO2. So if blood PCO2 levels rise, this is called hypercapnia. So if they rise, CO2 levels accumulate in the brain, and it's going to join with water and become carbonic acid. So if you remember back from when we talked about the carbonic acid and dissociating and all that, the next part's going to make sense. So carbonic acid is going to dissociate, releasing a hydrogen ion, and this is going to cause a drop in pH. This increased hydrogen ion concentration is going to stimulate central chemoreceptors of the brainstem, which synapse with respiratory regulatory centers. And then the respiratory centers are going to then, at that point, increase the depth and rate of breathing to lower PCO2, and the pH levels will return to normal. If PCO2 levels decrease, respiration may become slow and shallow, or become slow and shallow. And then this is where we're going to introduce the term apnea. And this is where breathing stops. And this may occur when PCO2 levels drop abnormally low. Now, sometimes what swimmers do is they voluntarily hyperventilate to help them hold their breath longer. What this does is it causes a drop in PCO2, and which is going to cause a delay in respiration because PCO2 levels are going to be needed to build back up. Unfortunately, this can cause dangerous drops in PO2 levels, so it's not a good idea. Next, we talk about the influence of PO2. So we have peripheral chemoreceptors in the aortic and carotid bodies that sense arterial O2 levels. Declining PO2 normally has only a slight effect on ventilation because of huge oxygen reservoir bound to hemoglobin. And this is just showing the location of the carotid body here and the aortic bodies here. And then we have the influence of arterial pH. So pH can modify respiratory rate and rhythm even if CO2 and O2 levels are normal. And this is going to be mediated by peripheral chemoreceptors. If the person has a decreased pH, this may reflect a number of things, including CO2 retention, accumulation of lactic acid, or excess ketone bodies. The respiratory system controls attempt to raise pH by increasing respiratory rate and depth. Here's a summary of the chemical factors. So the first is that rising CO2 levels are the most powerful respiratory stimulant. Normally, blood PO2 levels affect breathing only indirectly by influencing peripheral chemoreceptor sensitivity changes in PCO2. I'm just going to skip that one, and then changes, changes in arterial pH resulting from CO2 retention or metabolic factors act indirectly through peripheral chemoreceptors. We also have influence of higher brain centers. We have hypothalamic controls that act through the limbic system to modify the rate and depth of respiration. As an example, a person can hold their breath in response to anger or gasping with pain. And changes in body temperature also change the rate of respiration. There's also some cortical controls, direct signals from the cerebral motor cortex that bypass the medullary controls. And this just means that you can voluntarily hold your breath until the brainstem reestablishes breathing when the blood CO2 levels become critical. There's also pulmonary irritant reflexes. There's receptors in the bronchioles that respond to irritants like dust, accumulated mucus, or noxious fumes. Promotes a reflexive constriction of air passages. And then some irritants can trigger a cough in the trachea or bronchi or a sneeze in the nasal cavity. And then finally, we have the inflation reflex. So stretch receptors in the pleurae and airways are stimulated by lung inflation. And what this does is when the lung inflates, so everything is kind of stretching out, it's going to send an inhibitory signal to the medullary respiratory center to end inhalation and to allow the person to exhale. And this may be a protective response more than a normal regulatory mechanism. So this is just showing all the different things. We start off with the respiratory centers and the medulla and the pons. Higher brain centers, we have the cerebral cortex. You can voluntarily control or hold your breath until CO2 levels get too high. We have the hypothalamus. You can hold your breath kind of in response to anger or gasping with pain. Oh, here we go with the cursor again. Goodness. Peripheral chemoreceptors detect decreases in O2, increases in CO2, and increases in hydrogen ion concentration. We have the stretch receptors in the lungs, the inflation reflex, and then the irritant responses. So in the cough, the trachea and the bronchi, you can end up getting coughing, or nasal cavity can cause a person to sneeze. Here is the question. And that is all I have for you for this talk.